All right, so we are going to start today and make the shift from Mendelian genetics, which is the study of heredity and inheritance, and how those traits are inherited, to actually looking at the components that allow us to inherit traits, which is our DNA. So this branch is actually called molecular genetics because it has to do with how we make the molecules that help to keep us alive. So let's take a look quickly here at what exactly is DNA. The basic building block of DNA is a monomer called a nucleotide. And this nucleotide has three parts. A sugar, which is deoxyribose, and that's the D in the DNA. The phosphate group, which is designated as a PO4, and a nitrogenous base. So this is kind of like a simplified version of what this looks like. So we've got the sugar here, and that little circle there is actually an oxygen. Then we have the phosphate and the nitrogenous base. So how we actually identify what nucleotide we have is based on this nitrogenous base. The phosphate and the pentose sugar, the deoxyribose, are always the same. It's the nitrogenous base that is different. So in DNA we have four different nitrogenous bases. We have adenine, we have thymine, we have guanine, and we have cytosine. So what we do is we take these pieces and we arrange them into the double helix structure of DNA. So how we arrange this is the sugar and phosphate groups go down kind of the sides of the ladder, and remember the ladder ends up being twisted. So you have an alternating sugar, phosphate, sugar, phosphate, sugar, phosphate down the sides. The nitrogenous bases, each side has a nitrogenous base that kind of sticks into the middle. That would be like the rungs or the steps of the ladder. And how they're held together is a hydrogen bond. So this is basically like almost like a magnetic force that holds them together. So, excuse me. This is kind of what I was talking about. So we've got one set of nucleotides on the left, another set of nucleotides on the right. So we go sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, all the way down on both sides. The nitrogenous bases line up across from each other, and you can see here we have hydrogen bonds. Now you'll also notice if you look here that there is a pattern, and I know you learned this in biology last year, where I have the A, which is adenine, always is paired up with a thymine, and if you check you'll see that. And then the G, the guanine, is always paired up with the cytosine. So this is what we call the base pairing rule. So before cell division can happen, one of the things that we have to do is copy the DNA, and this is referred to as replication. So the structure allows us to explain how replication takes place, because basically if you unhook the nucleotides from each other and kind of split the chain in half, so if I kind of split it down the middle here, I can actually create the other side using the left side as a pattern to create a new right side, and using the right side as a pattern to create a new left. And that's what we call a template. A template is just simply a pattern. So the idea is if I have one side that has a code of A, T, G, C, T, T, G, G, A, T, C, A, then I can actually create the other side. And I can do this using this pattern. So we know that the A is going to pair up with a T, and that the T is going to pair up with an A, and so on. So I want you to pause the video right now and come up with what the other side would be based on the base pairing rule. Okay, so now you're back, and let's check it. So the other side will automatically be this. So check your answers and make sure that you did it correctly. Now remember, this is when we're using the existing DNA to create the new DNA, and that's important. We can't use this would not be if we're creating something else. All right, so you want to flip over to your handouts packet, and you should have this in there. All right, so the directions at the top say 4 nucleotides make up DNA: adenine, cytosine, guanine, and thymine. These nucleotides always occur in pairs called base pairs. So you're going to write the missing letter to create each base pair, and the first 2 have been done for you. All right, so at the top there you can see we've got our C paired with our G, and we have our A paired with our T. So the next one here we have G, so that will pair with C. Another G pairs with C. C pairs with G. C pairs with G. T pairs with A pairs with T, and C pairs with G on the way down. All right, so with the questions at the bottom. All right, what nucleotide is always paired with thymine? That is adenine. And what nucleotide is always paired with guanine? That is cytosine. Okay. All right, so that kind of completes our practice here with DNA replication and talking about the structure of it.